Good evening to you all. Thank you for tuning in to tonight's Seaweek Ngā Kōrero event, our underwater forests. We ran a wonderful series last year. We've had a little hiatus, but tonight we are back to learn about the significance of seaweed. I'm Alison Balance. I'm your MC for the evening. I'm a natural history writer, and I'm a big fan of all those things out in the big blue, but also the green things, the brown things, and the red things, which together make up our wonderful seaweed. And I'm also a very big fan of seagrass, which I see on the estuary out my window. With me, virtually, we have three fabulous guests. I'll introduce them very soon, but first, let's have a karakia to start with. Tukua te wairoa kia reri ki ngā taumata. Hai arahi i a tātou mahi. Me tā tātou whaia i ngā tikanga a rātou mā. Kia mau, kia ita, kia kore ai ngaro, kia pupuri, kia whakamoa, kia tina, hui e, tai ki e. Kia ora again. If you're new to this, a very big hello. If you've joined us before, welcome back. You'll know how this works. We'd love for you to introduce yourselves to us in the chat. We love hearing from you and learning where you're tuning in from. I'm at the top of the South Island in lovely sunny Nelson, although it may be wet here tomorrow. The chat box to do all of this is accessed via the text bubble icon at the bottom of your screen, which should bring up the chat panel on the right-hand side. A bit of background about Sea Week, Kaupapa Moana. It is New Zealand's annual national week, celebrating the sea, Te Moana, and it's coordinated by the Sir Peter Blake Marine Education and Recreation Centre. So a big ups to them. Sea Week's pinnacle of events occurs annually at the beginning of March, so we've missed it for this year. But Sea Week has a year-round mission, which is about exciting and inspiring all New Zealanders to renew their connections with the sea. That's why Sea Week is hosting this Ngā Kōrero webinar series, exploring ocean kaitiakitanga in Aotearoa, New Zealand. Throughout the Ngā Kōrero webinar series, we have put the spotlight on many different topics. We're doing that to emphasize the diverse connections and interactions that we all have with the sea. It's a good reminder that our daily lives are intertwined with the health of our ocean environment. A huge thanks to our long-time supporters of Sea Week, and in particular, those that have made this webinar series possible, the New Zealand National Commission for UNESCO. Kia ora, thank you very much. In previous webinars, we've covered all sorts of things: the downstream legacy of sediments and nutrient pollution, how we look after kai moana to ensure there is fish for generations to come, the problem with microplastics. We've celebrated our magnificent marine megafauna, and we've talked about the changing climate. Now, if you've missed any of these, they are all recorded and you can watch them anytime on the Sea Week YouTube channel. And this webinar is also being recorded so you can watch it again or share it with others. Now, I am very excited to properly introduce tonight's amazing lineup. First of all, we have Dr. Blain. Caitlin's a postdoctoral fellow at the University of Auckland's Leigh Marine Laboratory, right next to the wonderful Goat Island Marine Reserve. Caitlin has studied how the primary production of canopy-forming macroalgae, that's the really big stuff like the trees of the underworld, water world, how they're impacted by the stuff that we humans do on land. She'll be followed by Zoe Studd. Zoe is the co-founder and director of the Mountains to Sea Wellington Trust, which works to connect people to te taio, to nature across the greater Wellington region. And since 2020, she has also led the Love Remurumu project, which aims to regenerate Wellington's underwater forests. Our final speaker will be Dan Crossett. Dan is a marine ecologist. He's part of the Cawthorne Institute's restoration team here in Nelson, and he focuses on the state of environment monitoring and bottom-up ecological activities that may enhance recruitment of foundation species I hope he tells us a bit more about that. After all the speakers have presented, I will ask them your questions. You can send us your questions at any time using the Q&A function, which is also on the bottom of your screen. So let us get to the real stuff of the evening. Starting us off tonight is Dr. Caitlin Blain. So Caitlin's from Leigh Marine Laboratory. She's currently investigating the role of kelp forests in coastal carbon cycles and climate change mitigation. Prior to this, Caitlin completed her MSc in marine biology from Memorial University of Newfoundland over in Canada, where she focused on the ecology of grazing-resistant macroalgae and their associated epifauna. Looking forward to hearing more, Caitlin. Take it away.
Thanks, Alison, and thank you, Sarah, as well, for setting up this great talk tonight. I'm looking forward to hearing from the other panelists today. So hello, everyone. Kia ora. I'm Caitlin Blain. As I've already been introduced, I'm a postdoc at the Leigh Marine Lab for the University of Auckland up in by the Goat Island Marine Reserve. And today I'm going to try and do a very quick overview of valuing kelp forests with focus on some of the research that we've been doing in the lab, and that's specific to New Zealand. So just as a really quick overview, I'm going to talk about sort of kelp services and how do we value kelp forests, and specifically how we understand the contribution of kelp forests to carbon sequestration, which has kind of been a big topic in lots of research fields and news and up and coming. And then looking at how restoration and efforts into saving our kelp forests is going to help with securing these valuable services that they provide. So kelp forests, and when I say kelp, I mean pretty much any of the large kelp and fucoids, which are these canopy-forming seaweeds from brown algae that are distributed across the globe in temperate regions and cover approximately a quarter to a third of our coastline. So they have substantial coverage, and they're also highly productive ecosystems, so as comparable to any terrestrial forest that we have. And they're significant components of the ecosystem. They are habitat modifiers, so they create shade and wave attenuation. They're incredibly important for the chemical modifications that they make to their surroundings. For example, when they take in nutrients and photosynthesize and release dissolved organic carbon and oxygen. They also mitigate changes in pH and create nice local buffers for some of the marine life that live within the kelp forest. And this is just an example of how kelp can sort of help change the pH environment around them. They're also really valuable in terms of how they create space for biodiversity, increasing ecosystem function, again, providing that habitat and nursery for many different species, which is really important for our fisheries and tourism and just the health of our coastlines. And like I've said previously, they're very efficient carbon fixers. They're very good at primary production, and this leads to ongoing benefits up the trophic line and secondary production. And they're gaining more attention for their potential as sort of a blue carbon donor, potentially contributing to carbon sequestration, which we'll get into in a little bit. I'm just putting up here because one of the things that has been sort of limiting kelp forests as being thought of as a blue carbon ecosystem is the fact that they have a high turnover, so they grow really fast, but that tissue that is growing is continually being released into the system. And they also don't grow in a soil or soft sediment. So they have a relatively small, what we call standing stock, so living biomass compared to other ecosystems. And this was a study that did this comparison of ecosystems in the UK. And you can see that kelp, compared to things like seagrass and tidal marshes, they have a relatively low standing stock. But we also know that the carbon flow through kelp forests is exceptionally high. So one of the highest compared to any of the other ecosystems. And so part of the challenge in valuing these kelp forests, in particular in that carbon sequestration realm, is figuring out what happens to that carbon once it's released from the kelp forest. So how do we value kelp forests? And maybe another question is, why should we value kelp forests? And these are sort of our unseen underwater forests, as it was put a little bit earlier. And a lot of people just sort of forget about them. If you don't see them, then you don't really realize the value that they have. But when you walk through a terrestrial forest, you dive through a kelp forest, you can really see the value with your own eyes, the species that they support and all the benefits that they provide to the ecosystem. And unless we provide some kind of value to the forest, then that really inhibits our capacity to help to conserve and restore and put forward initiatives to make sure that our kelp forests are staying healthy and staying abundant. So there's lots of different ways that we can put values on any of our marine ecosystems, kelp forests included. And I'd like to start by pointing out that when we value an ecosystem, it's what humans get from that ecosystem. It doesn't speak to the value of the ecosystem in itself and what it provides to the different species in that system. But some of the things that we obviously value ecosystems for, particularly kelp, are what we can harvest from them, the primary production that they have, cultural and social benefits that we get from the kelp forests, tourism, for example, and climate control, and any kind of disturbance mitigation like storm amelioration or wave action amelioration. So regionally, there have been many different assessments that have looked at how we can value kelp forests and what are important aspects that we need to include when we're valuing the ecosystems. And one that I was really lucky to be recently a part of is a global study where we came together with a group of kelp forest ecologists and pulled together a global data set for the first time that combined three sort of main factors, main services that kelp forests provide to us. And those were fisheries production, nutrient cycling, and carbon dioxide removal. And I'd like to point out again, these are just three different services. And obviously kelp forests provide a wealth of different services that are really hard to account for in one study. And also we only were able to include 6 different kelp species, sort of the main kelp genre, sorry. And here's just a little summary of the paper that came out recently. And it shows a bit of the distribution and then how they're valued regionally based on just those 3 different factors. And what it came out to is that we see that kelp forests can be up $500 billion plus, US dollars plus, in revenue per year, just based on those 3 factors alone. And what struck me as really interesting in the study is that nutrient removal was the top sort of valued service. Fisheries production was the second, and carbon removal was actually the third. And I think what that speaks to is our societal valuing of carbon at the moment, and that it's not probably as high as it needs to be in terms of trying to mitigate those CO2 emissions. So that was a really interesting point that came out in this study. And again, it was just a really nice exercise to show that if we put these dollar signs on kelp forests, while it's not going to be 100% accurate because we're missing some services and some species of kelp, it gives those policymakers and those people wanting to get involved, it places a value on the ecosystem that they can say, look, this needs more attention. Kelp forests are probably one of the ecosystems that receives some of the least funding research-wise and conservation-wise when we compare it to coral reefs and other systems. And so we do need more attention on kelp forests because unfortunately they are in a sort of overall global decline. And so this was just the highlight of the study that we found. And obviously, kelp forests have, again, been receiving a lot of attention for their capacity to potentially mitigate some of the impacts of climate change, whether it be through buffering pH changes in the water for ocean acidification or for sequestering carbon dioxide. And I just put this up here. It was just a quick search just to show you how research on blue carbon is a really developing field. We still don't know nearly enough about it. And so that we're just keep building and building and building on these knowledge bases. And you can see here in the green and the yellow at the bottom parts of the graph here, in terms of other blue carbon ecosystems, we really don't know that much about how macroalgae can contribute as a blue carbon donor or ecosystem. And a lot of this revolves around the fact that accounting for carbon, as I mentioned earlier, in marine ecosystems is very complex. So this really nice summary of this was published by Katrina Hurd in 2022, and basically just demonstrating that in a terrestrial system, we have a relatively straightforward accounting in that a tree holds a lot of carbon in it. And as its litter falls, it gets incorporated into the soil and it can be held in there for a long time. In a marine coastal system, we've got a lot of interacting variables. We've got wave action, we've got tidal action, we've got a lot of grazers that are eating up that carbon and distributing it in other places. So there's a lot of things that are going on. The kelp is turning over its tissue really quickly, and it's just releasing it back into the ecosystem. So we need to sort of answer some of these questions about where that carbon goes before we can know how valuable kelp is to us on that blue carbon stage. And that's where a bit of my current work has been looking into is, can kelp forests mitigate CO2 emissions? And this is a multi-step question, and there's many aspects of this question that are targeting that complexity, but I'm just biting off a small piece of that to sort of answer. Starting with, we need to know how much carbon is actually released from the kelp and what form it takes when it's being released. Is it full tissues of the kelp? Is it small particulate matter? Is it a dissolved organic carbon? All these things contribute to how far the carbon will go in the ocean, how long it will stay in that form. And then another question is, again, that longevity in the fate. So where does it end up? Does it get stored for a period of time that's significant enough to make a difference to CO2 in our atmosphere? So first, I'll just focus on this first question. And we've used several different methods to get out the primary production of kelp forests and looking at how this varies across environmental gradients in northeastern New Zealand. And one of the biggest stressors that we have in the Hauraki Gulf is sedimentation and turbidity. As you get close to the city of Auckland, we've got a lot of turbidity in their water, which blocks out light, which is an essential for our primary producers, as they need light to photosynthesize. So when we look at kelp forests in our most pristine sites, we see that these kelp are highly productive as the giant kelp forests of North America, as productive as any rainforest terrestrially. And there's a good division in how the carbon is being released from the kelp. So about 40% of that is released just by sloughing of the tissues into a particulate matter. And about 40% is released as what we call that dissolved organic carbon. So that's carbon that's basically fixed from the environment during photosynthesis, but it's not incorporated into the plant when it grows. And so it's released back into the environment. When we go to our low light sites, we get a significant reduction in the abundance of kelp, but also a significant reduction in how much they're fixing carbon, which is demonstrated by the size difference in these circles here. And we can also see that we're losing a lot of our dissolved organic carbon production. And again, this difference in the form that the carbon is being released from the kelp may have some quite significant implications into how valuable those kelp forests are for sequestration long term. So the next question then follows, where does it go? And I've done this really quick schematic just to give you an overview here. So we've got some little kelp on a reef, they're photosynthesizing, they're bringing down CO2, hopefully it's drawing CO2 down from the atmosphere. And the thought is that about 80% of the particulate matter, so tissues from the kelp and little particulates that come off as it's growing, that gets exported into neighboring ecosystems. And An even smaller amount may make it to the deep sea, which is an environment where long-term sequestration can happen more efficiently because there aren't as many of the little microbes, and there's not a suitable environment for that carbon to break down back into carbon dioxide. But as my work has showed, there's actually a high proportion of carbon that's released in a dissolved form. And we actually have some evidence from previous research that suggests that this dissolved form is actually a really long-lived form of carbon compared to other forms. And we know very, very little about this dissolved form. And I'll just put up, skip here. This little graph here, it shows you most of the work that's been done on kelp really focuses on this POM. So that's that particulate organic matter rather than dissolved organic matter. So it's an area that we really need to know a lot more about. Okay, so to start getting at these questions, we've been doing some degradation experiments in the lab and in situ and across different environmental gradients in the Hauraki Gulf. And looking at how both the tissue of the kelp and the dissolved organic carbon of the kelp degrade and how quickly they degrade. And what happens is that when we get a curve like this, where there's a bit of a stabling out at the bottom, suggests that some of that kelp, some of that carbon that's released from the kelp is long-lived. It reaches a point where it doesn't degrade anymore, and that carbon may be quite impactful for carbon sequestration. What was also interesting to find in studies where we were looking at the degradation of the tissue is that Eclonia compared to other, Eclonia radiata is the kelp that we have in eastern New Zealand. It's the dominant canopy-forming species. And what we found is that it degrades quite quickly compared to a lot of other species in the literature, at least its particulate form. And so this may mean that it's potentially not as long-lived the carbon as other species. Then if we compare these to how the dissolved form of carbon degrades, you can see a kind of stark contrast here that the dissolved form may be, as we expected, potentially more long-lived. So instead of only maybe less than 10% of the carbon remaining in the system, after even almost 100 days, we get closer to 40% or more staying in the system. So this longevity of the carbon is really important for carbon accounting. But there are still a ton of things that are unknowns and research that is still ongoing and developing so that we can help to put in the data that is going to help us implement strategies for accounting for carbon sequestration from kelp forests, and how can we integrate that into management plans going forward. So everything from looking at tissue degradation to dock degradation, what's actually making up those different particulates and dissolved organic carbon, and also looking at how kelp forests act as buffers within the system with pH and nutrient amelioration and those sort of ecosystem functions. And this is all really important to get at, because actually kelp forests are facing their own sort of global stressors. Regionally, the abundance of kelp varies, and the stressors that impact kelp vary across different regions around the globe. But as a whole, we are seeing a net decline in kelp forests every year. And I think it's something about 2% loss per year. There will be some areas that get more kelp and some that get less, but as a whole around the globe, it's a net decline. And so it's important that we understand how to value them now and how they function now, so that we can really harness in on getting them integrated into management plans. And there are a number of initiatives. I'd like to point out the one, the Kelp Forest Challenge, has been put together by the Kelp Forest Alliance, which is a global network of kelp forest ecologists that working together to find ways to promote the restoration and conservation of kelp forests. And they've just released their targets, which are very hefty, I believe, 4 million hectares of protected kelp forests by the year 2040. And that lines up with other initiatives such as the one I've got on my screen here, the 2022 UN Biodiversity Conference, which was held in Montreal, and their goals of reaching 30% protection by 2030. And so people can get involved in this. This is not just for researchers. This is for anybody who has interest in kelp. It's for governments, it's for politicians, it's for schools, it's for community groups, universities. All sorts of people can get involved in this in small ways or big ways. If you go onto that, I should have put the link there for you, sorry, but if you Google them and you look on their website, you'll see how many people across the globe have actually already started various restoration projects to help kelp forests to keep them protected, but then also to restore the ones that have been lost. So restoration, I'm just going to do a really quick couple slides on this, because I know Kelsey Miller is speaking at another one of these events, and she will talk about this more. And I think Dan will probably talk about a bit of this as well. And restoration of kelp forest, so trying to bring back kelp forest where it has been lost, it's really important to understand the root cause of the loss of the kelp forest in the first place. It's not going to be the same across even a country or a region. I know some of the main reasons that we've lost kelp forests in northeastern New Zealand are very different than maybe some of the reasons they've lost kelp forests in southern parts of New Zealand. And so having an understanding of what the cause of the kelp loss was originally and how the environment has changed is really important for making informed decisions about how you then restore and manage that ecosystem. And so there's a number of different ways, and this figure sort of points out some of the major ones about transplanting, reseeding, doing genetics to rear some, for example, thermally tolerant species, removing herbivores or restoring ecosystems using protection. So there's lots of different ways. And I thought I'd just touch briefly on an example from northeastern New Zealand, where we have extensive kinna barrens in a certain depth distribution in New Zealand. And this is primarily caused in the Hauraki Gulf by the overfishing of the main predators of kinna, so snapper and crayfish. And when there's not enough of these large predators in the system, we get an overabundance of kinna, and the kinna mow down the kelp forest. Kinna are not a bad species. Kinna are an important part of a functioning, healthy ecosystem. But when the ecosystem's out of balance, it becomes a bit of an issue. And so we've actually had a student, Kelsey Miller, who's been working on a project to look at how experimentally we can manage the kinna populations to help sort of jumpstart restoration initiatives such as marine protection, for example, which is marine protection is a passive sort of way of restoring the kelp forest by not allowing fishing, letting the predators come back into the system to control the sea urchins, and then using something like a kinna removal strategy or harvesting strategy to lower those numbers to help jumpstart that restoration. And she's done some really amazing work across 4 sites in the Hauraki Gulf, and looking at how we take down kinna numbers. It's really important work that she's done, and a lot of thought went into planning and the sites, the specifics of the locations where we did remove the kinna, what was done with the kinna, how we went about lowering their numbers, and sorry, I'm getting a bit of a tickle in my throat now. And the monitoring that went on alongside that to see how the kelp went by. And it's been about 2 years since she's gone to sites and significantly lowered the kinna numbers at sites that we're looking at an area of about 1.5 to 2 hectares for each of the sites. And the positives of this is that the kelp forests come back, and they come back rapidly. And why that happens in northeastern New Zealand is because We know that one of the main stressors there is the kinna. It's not temperature. It's in this particular spot, it's not sedimentation. And we also know that there's deep kelp forest that can help to reseed those reefs naturally. So that's been really positive. But one of the things that it doesn't do is restore the ecosystem completely. So we still don't get those predators coming back. We still don't get a lot of the fish coming back because these are still heavily fished areas. The kinna eventually will come back in, and we are starting to see that after 2 years. They eventually will come back in, and we will still have a barren unless there is active sort of management or protection that's put in place on top of that. So this was a really nice figure that she put together for her one of her papers that kind of goes through the efforts, the money, and the efficiency of these different sort of management strategies. So and ultimately, I think again that she's found that a combined approach where you're addressing the cause of the barren, so the loss of the predators, and then using some kind of active restoration strategy to help jumpstart restoration is what was going to work best in the end. So what's next? There's still a lot of work to be done on understanding the role of kelp in carbon sequestration. As I've outlined before, like kelp carbon longevity, where does it go? Does it end up in the soft sediments? How long does it stay there for? And what impacts are we going to expect with some of the stressors we've got, such as that sedimentation and increased storm events, warming? We have really limited ideas about how climate change is going to impact our kelp forest productivity and also how they contribute to those coastal carbon cycles. We also know far too little about other ecosystem services, which I'd mentioned earlier, how they contribute to fisheries and nurseries for other species and nutrient cycling. And understanding the extent and drivers of kelp loss in Aotearoa, it seems like it would be a basic question that we know really easily, but it's actually quite complex. And so we need to know more about that before we can get these strategies in place for restoration. And so there's a lot for us still to learn. We know a lot, but there's still a lot for us to learn as we go forward with trying to protect our forests and the services they provide for us. So I don't know how long I've been talking for, but I'll stop there. But I just want to say a really big thank you to everyone in the NZ Reefs Lab with Dr. Nick Shears and Live Ocean Foundation for supporting me, and the George Mason Center for the natural environment. So thanks very much.
Kia ora. Thanks heaps, Caitlin. I learned a lot in that. I'll just share something that Leigh has put in the chat that you may or may not have seen in response to Caitlin talking about removing kinna to help the kelp come back. She said the great thing is that the Waiheke Marine Project have been doing this for a couple of years, working with locals and mana whenua. And then she also mentioned that rahui is another option iwi are managing, and that is more around what can be harvested and collected. So it will be interesting to see where that goes in future. So thank you.
Absolutely. Yeah.
And if you have any questions, listeners, please ask away in Q&A, and we'll ask them after all of the speakers have presented. Now, next speaker is the lovely Zoe Studd. So Zoe's work and studies have always been connected to the marine environment. She started as a dive instructor, and she had studies in environmental marine science in Australia. She apparently worked briefly as a classroom teacher, but quickly moved into outdoor environmental education. So Zoe is the co-founder and director of the Mountains to Sea Wellington Trust. And since 2020, she has also led the Love Rimurimu project, which aims to regenerate Wellington's underwater forests, so Wellington, not the Hauraki Gulf, and in doing so, inspire the community to become involved in the project. Tell us more, Zoe.
Maria Koto. Thanks so much, Alison and Sarah, for organising this event to talk about all things seaweedy and nerdy. And Caitlin, loved listening to your kōrero as well. And yeah, everything comes together. So it's easy, I think, to get lots of seaweed nerds raving. So I'm going to work quite hard to stick to my time limit as well. But So yes, my name is Zoe, and I'm have been working in freshwater and marine education and restoration for over a decade now, and am part of an organisation called Mountains to Sea Wellington. We work across freshwater and marine ecosystems. And yeah, several years ago, we started to get excited about seaweed. And at that stage, I don't think I knew the journey that was going to take us on, but here we are, and we've been really enjoying it and learning a huge amount along the way as well. So tonight I'm going to talk a little bit about how we started to also share our love of rimu with a wider community, to get them involved in thinking about what was actually happening under the ocean, and our journey on towards regeneration of some of Wellington Harbour's lost seaweed forests as well. So it's a pleasure to be here. Now let me just make sure I can move on. There we go. Beautiful. So for us, when we began this project, and with all of the work at Mountains to Sea, we start with connection. So we start with community at the heart. So how do you encourage a group of people who may not want to enter the ocean, or who do enter the ocean but find seaweed really slimy, frightening, smelly, and generally something that they don't want to get involved with? And, you know, we've all come out on a Thursday night to talk about seaweed, but most people, that's not necessarily a naturally interesting topic to them. And I think there's a sea change in New Zealand. I think people are becoming more and more interested at the potential and the opportunities ahead. But for us, when we got underway, we were like, right, it's got a PR problem. How are we going to engage people in what we want to do? So we've always run education programs, and we developed a special education program called Love Rimu. And that began a really wide collaboration. So researchers here from Niwa and Victoria, from Mana Whenua Taranaki Wānui, and we worked with 3 kura kaupapa Māori in that first year, and some mainstream kura as well. We had cocktail events, we sold t-shirts, so we drank it, we ate it, we rolled around in it, we held community snorkel days. We got people to really kind of come down and start to engage with it. And that was a really wonderful process, because a lot of people who'd never really noticed what seaweed was started to really kind of, you know, get in there and start sort of getting to know their local seaweeds a bit better. And you touched on this as well, Caitlin, but one of the stories that, one of the sort of things that we like to share with people is this idea that our ocean in the forest is exactly like our ocean on land, our forests on land. But our forests on land get all this attention. You know, everyone's involved in the pest trapping project. Everyone's got, you know, maybe a little area down the road that they're able to go and help planting at. And it's much more difficult for community to be connected in that way in the marine environment, but they're just as important. So there's, when people go out snorkeling, they'll know, or if they go in the forest, you know, they'll know the fronds of the rimu tree. And when you snorkel, there's also the fronds of the rimu, the seaweed forest, and they're almost identical. There's these beautiful kind of filaments, these green filaments. So understanding those connections and all of that importance starts to kind of pique people's interest and invite them into that, into those conversations. I won't touch on this because Caitlin has more than covered it, and spends probably a lot more time thinking about it. It's really fascinating to hear about your work. So for us in New Zealand, for us in Wellington, another one of the key drivers was this is so up north they don't get Macrocystis porifera, this giant kelp. It's absolutely beautiful, can get to these really great heights, and it's essentially sort of swimming through a massive seaweed forest if it's nice and healthy. But in Wellington Harbour, we're at our northernmost limit. It doesn't grow much further north than Wellington, and we're starting to see the decline. We've already seen the decline of this giant kelp species in the Wellington Harbour. There's lots of different drivers for that, but it is a species that is threatened, and we're really concerned that we're going to lose it from Wellington Harbour, and with that we're going to lose a lot of those ecosystem services as well. And as people have touched on, climate change is a large driver, but for us sediment is another one of those, really significant impacts that we see every time it rains. In Wellington, we'll see, a plume of sediment move across the harbour. And that's essentially, for young seaweeds, it's like having a bit of sandpaper passed over the top, but it also blocks light, which really inhibits their growth as well. Overfishing and all those associated cascade effects and water pollution as well. So in Te Whanganui Tara and in the Great Harbour of Tara in Wellington, we are looking at all these cumulative impacts. And over time, we know that there's been a decline, but it's not quantified. There's like some bits of research that have been done here and there, but when we started trying to investigate, we really didn't know what the state of decline was, how much had actually been lost from inside the harbour. So we started to kind of collect a bit of information and to start to do some baselines and chat with community about what they'd noticed in terms of the changes over time in their harbour. And the picture was pretty bleak, right? Like we've got several really important islands in the middle of the harbour, Makoro, Ngā Mokopuna, Makoro, and Machu. And most of them had lost all the seaweed cover around them. And also that's reflected through lots of other parts of the harbour as well. So the way that our program grew, in terms of working into regeneration here in Wellington Harbour was very community-led, and it actually came from the young people. It came from the students that we've been working with, and in particular from Te Kura Kaupapa Māori, Ngā Mokopuna, Te Kura Kaupapa Māori o Ngā Mokopuna, who are in Motukairangi on the Miramar Peninsula, and Te Arawhanui. And by year 2 of the project, I don't know that they were entirely convinced at the beginning why we were going to be doing a whole year on seaweed, but by the end of it, I think they thought it was quite a good time. But as we moved through the program, we moved into mapping, and they actually produced the first proposed restoration sites after going through a mapping exercise, talking with mana whenua about sites of cultural significance, looking at, where freshwater came into the harbour, looking at some of the old sediment flow maps we could find. And they came up with their first proposals for restoration and presented those back to mana whenua. And from those seeds, from those sort of 2 years through the education program, we actually applied for some funding through the Wellington Community Fund, and that began the regeneration project in 2021. And we stay, I think, really true to the kaupapa that community need to remain really connected with the project and really help to champion these kinds of projects, because it's a disconnection from an environment that allows it to degrade out of sight and out of mind. So we think of every opportunity to make sure people continue that connection. And the lovely Murimu program continues on in schools, and we still do lots of stuff through our restoration project as well and with the community. But now, much more so, we have greater guidance from mana whenua Taranaki Wānui, and also working to support their moemoeā for the harbour. So their aspirations for the harbour, seaweed regeneration is but one part of what they would like to see in terms of the restoration of the Māori of their harbour as well. It's informed, so the project is informed by research, mātauranga and community science collaborations. There's bits of research going on, but some of it as well is looking into the thermal tolerance of those macroalgae species that we're worried we're going to lose inside the harbour as well. We also work in a really open source way and really collaboratively, and that's so that we can share what we're doing as we're going on this journey, because there is so much to learn, and we're also prepared to make some mistakes and to give things a go. And I think there's a freedom in being a community organisation doing that sometimes, to be able to kind of be a little bit more experimentative in some of our approaches. So in terms of growing a seaweed forest, I don't know if I should include this, but for me, when I began, I didn't actually really understand the process of how you grew a seaweed forest. We said, yes, students, you really want to grow some seaweed, we'll help you. And then we went, how do we help you? And actually, it was kind of a paucity of information for a while in this space. And there's lots of people now starting to work in it more and more in a community gathering around restoration, and that's just fantastic. But very simply, there's this collection of fertile tissue from the wild, and that's really interesting, just kind of noticing those darker patches or those raised patches on the blades of seaweeds. they get then taken to the hatchery at Niwa, and they, poor things, they get dried out and then they get put in the fridge and in the dark for a night to get cold and dark, and then they get put back in the water, and that releases the spores, the sori, the spores. And after a period of time, they develop into sporophytes, are small seaweeds. And there you can see a little photo. I don't know if my pointer's on it, but this little photo here, this is a restoration technique, and Caitlin talked about it as well, called green gravel, which is seeding rocks that can then go back out into the marine environment. And where we are at the moment is sort of in this phase here. So we have some out planted into a pilot site just in Mahanga Bay, but this winter we are moving into going into some more restoration sites. And then we'll be moving into monitoring how our seaweeds go in those different areas. So this just kind of shows some of the things that's going on in that hatchery to nursery phase. So that's out in the field, collecting some of those, some of the sporophytes here, also the sori. This is the sori here, and collecting the spores. This is Roberta D'Acino, who we work with at Niwa, and our macroalgae technician. And you can see some of the early days of trying out, different substrates for growing our seaweeds on as well. And we've been doing a lot of trials with different substrates, including now with Taranaki Wānui, looking at traditional fibres that might have been used for other restoration or translocation of other species, and if they can also be used as substrates for planting out into the marine environment, because we don't want to perpetuate putting out a whole lot of plastics or other things that might degrade in the marine environment as well. Some of these different techniques for us, probably the two that we'll use this year will be this reseeding of our green gravel. So using rocks that have been reseeded and have the young seaweeds on them, and also some of these aquaculture lines. And we've got quite a few challenges ahead of us. We have kinna in some of these sites, we have pulse events where we get large amounts of sediment as well. So we'll be doing sort of research alongside to see how our seaweeds survive. This is some of the growth in our pilot sites that got us all excited in the first year. We had about 90 metres of growth on some of those seaweeds, and then we actually lost quite a few with our incredibly warm summer. And that warm summer has also had a bit of an impact on the health and the viability of some of the spores that have been collected over the last few months. So by now, we would have expected them to be certainly a lot healthier than they are. But in the lab, when they're having a look at them, they're just not quite as healthy and as viable as we'd hope for by this time of year. One of the really fun things about seaweed is that everything happens, it appears that everything seems to happen in winter. So it means we have to spend a lot of time in the water in the middle of winter. And sometimes I kind of wish we'd gone for corals or something like that. And I understand why people get more funding to do that. So for us, our next steps with our regeneration sites, we have these 5 small-scale regeneration sites that will go in this winter. And that's been a really interesting process for us, because at the beginning, I didn't realise I was going to have to do a resource consent as well. And this, is actually another one of those barriers that community groups can face when they're looking at regeneration in the marine environment, the complexities of trying to move through the system that we currently have in place. Whereas if we had, say, a broader marine management plan, there might be opportunities to actually have regeneration areas where different kinds of regeneration could be trialled. But the nice thing about having gone through the consent process, which is still pending, is that then we can share that information and that process with other groups that are trying to do similar things in other places as well. There's also going to be some research sites that are going in, and some of those trials are going to look at kinda exclusion. So, setting up some of the same restoration techniques, but in caged areas so the kinda can't get in if they're a problem in a particular site as well. And, all of this is really working towards, and one of the outcomes from the project was this sort of a regeneration toolkit that can, share case studies and some of the processes and some of the techniques that we have trialled as well. And another really fun thing that has developed recently and has been another way to kind of engage and keep community engaged in the project and to have a strong sense of ownership is a free diving community monitoring project that we have been piloting called the Kelpers. And that's been partnering with freedive instructors and with some of the researchers here to develop a community monitoring toolkit. And the really cool thing is that some of the students from Ngā Mokopuna, who are now a little bit older, have also gone through that process. So they will be adjacent, their school is adjacent to one of those restoration sites. So they will be collecting the rocks, collecting the spores, watching them be, seeded and grow in the lab, and then helping with the plant out in one of those regeneration areas and then monitoring them as well. And to me, that's really that true connection with community with a regeneration activity is when they get to feel empowered throughout the entire process. We also joined up to the Kelp Forest Challenge, so I'm really pleased that Caitlin talked about that. We have pledged a hectare by 2030, and when you figure out how big that is, it's quite a lot of seaweed. So we've got a bit of work to do, but we're really hoping that we'll get there. We're also really hoping that some of the stuff that we learn along the way will help other organisations be able to do similar work in their area as well. But that's a really fantastic initiative, and to really galvanise efforts and awareness around the need for regeneration and to have the spotlight on our underwater forests as well. And collaboration has been key to all of this. We've had so many different people who've been involved. So I just really want to recognise all the community and the mana whenua who are involved with the project, and also our partners at NIWA and Victoria University and the people who've helped fund the project as well, which has been absolutely pivotal for us being able to continue on in this way. So, kia ora koutou.
Kia ora, thank you, Zoe. It's been lovely hearing about the project. I've heard about it when it started. I wasn't sure where you've got to with it. It's terrific. And I love the kelping hand.
You can come and kelp if you like, but it will be cold.
Okay, I'll be in touch about that. Anyway, thank you, Zoe. Now, don't forget to pop your questions into the Q&A tab at the bottom of the screen. We've got some questions coming in. We'd love some more. But before we get to your questions, we will get to our final speaker, the terrific Dr. Dan Crossett. So Dan is a marine ecologist. He's part of the Kōrero Institute restoration team, and he has spent the greater part of the last 5 years investigating large brown algal resilience and recovery following those big disturbance events such as the Kōrero earthquake, as well as more chronic stresses such as warming seas and sedimentation. And I also know that he does a bit of work on seagrass as well. So Dan, take it away.
Just getting down here. Thanks, Allison. Yeah, it was great to see you out on the seagrass meadow a couple weeks ago. That was pretty fun, huh?
Sure was. It was lovely out there.
Just working here. Sorry, everyone. I have a bit of a sickness I developed over the last few days with a 2-year-old running around, but hopefully it won't cause too much of an impact. So I'm Dan Crossett, and I'm going to tell you a little bit about what's up with large brown algae. Seaweed, everyone's kind of got a little more of an idea about this now because of Caitlin and Zoe's great talks. Kia ora for that. It makes my job a little easier. And before we move on any further, I just want to make sure I thank a lot of the people who have been pretty important in my journey here in New Zealand. I started at Vusil, actually, up near Zoe, and that's where I was when I first came over. And then since then, I moved to Kōrero and with Grant Hopkins, Donna Clark, and Paul South down here, and done my PhD through University of Canterbury with Dave Shield and Robin Dunmore as my supervisors. It's been an incredible 10 years over here. The last 5 years has been very informative, diving on the reefs of Kaikoura. And one of the big things that I will never forget was being welcomed onto the marae down in Kaikoura, which you can see here on the bottom image. And just really great way to just figure out everything that's happening at a local stance, and to be able to help out and to be a part of that has been really really great. And so we'll get into just a little bit of a rundown about large brown algae. Everyone probably already knows about this now that I'm the third speaker, but just a quick couple of points. So there's reds, greens, and browns, and I know Allison brought this up to begin with, but I think there's a distinction we just kind of have to figure out. And these large brown algae are part of the brown macroalgae, or and they're foundation species that create this habitat that Caitlin and Zoe were both talking about. They can shade, they can attenuate waves, they can decrease sedimentation, they can even change the temperature depending on how much light or seawater is coming in. They also provide refuge and food for a lot of important species such as paua and crayfish and a lot of the important fisheries that New Zealand and the world look after. And as Caitlin kind of touched upon a little bit, there's two major groups, and that's the kelps and the fucoids that make up these large brown algae. And so a lot of talk has been towards the kelp, but a lot of the work I've been doing is on fucoids because they're the ones who make up a lot of the shallow subtidal and intertidal zone around the Kōrero coast, particularly. And it's also really important. These primary producers or photosynthetic organisms are similar to terrestrial plants such as trees and stuff, and they drive these ecological currencies in these systems. So they capture light and take in CO2, as Caitlin was talking about, and convert it to energy, which then can be, harnessed up the system and make their way all the way to the big whales that are swimming off sea. So they're really important as an organism and as a life source to coastal environments. And just to kind of a quick shot here, I mean, Caitlin's already talked about it a bit, but they're massive primary producers. So they're in terms of in league with terrestrial forests and different phytoplanktons from different areas around the temperate region where they're really prevalent. They can double and triple and even quadruple some of these other productivity streams from different organisms. And so when we see them die off, or we see reduction in their abundance, it's quite alarming because they provide so many benefits to ecosystems and to human services, because that's a lot of human economy has been built to take advantage or use most of these systems. And kind of as we've touched upon here in the last hour or so, there's a couple of different ways they're being reduced in heat waves, global warming or warming seas, light attenuation, so that shading, that sedimentation that then causes light to be disrupted as it makes its way into the water column. Epiphany is a natural phenomenon, but when you have too many of them, such as henobarons, it'd be pretty dire. Sedimentation is particularly nasty in New Zealand. There's a lot of coastal runoff. This is just a natural thing as well from different rivers, but certainly there's been some anthropogenic effects to this. And just big disturbance events. So you have many times a major wave action event comes in, you get a lot of sediment, a lot of gravel that moves around and can smother things. One of the things that happens when you get all these accumulating stressors in one space is you get different environmental and biological gradients. One of the study sites I looked at was in Totanui in Queen Charlotte Sound. And here you have the inner Queen Charlotte Sound. You have a pretty degraded environment these days, and that's pretty low seawater mixing now, warmer seawater temperatures throughout the year, and a lot more environmental flux, seasonal flux. So you have a high algal blooms in summer, and then in the winter they all die off. And so you have a lot of this really interesting physical dynamics happening that changes quite dramatically through the year. And because of this, there's pretty low diversity and pretty low algal abundance in the inner Queen Charlotte Sound, compared to the outer Queen Charlotte Sound, which has higher seawater mixing, cooler seawater temperatures. It has a little lower flux because it's closer to the Cook Strait. And so because of this, you get a little more mixing happening. And it has higher algal density, but still pretty low compared to, say, Tory Channel, which you can kind of use a bit more as a control site, because over here there's similar temperatures in the outer Queen Charlotte Sound, but definitely more diversity, healthier environment. And you get this gradient that happens here is really important because you can kind of study the system and see what's happening. And we'll talk more about this later, but this is one of the case studies I looked at in my PhD. And then here's the big one that I've done a lot of my work with, and that was after the 7.8 meter or megawatt Kaikoura earthquake. that happened in 2016, and you've probably heard a lot about this, but you can see these pictures here where there was just massive devastation of subtidal reefs, intertidal reefs. All that brown is large brown algae that was lifted. This particular area is in Waipapa Bay, and there was 6 meters of uplift, so it's a lot. Over here on the left, I don't know if you guys know Professor Dave Shield, but he's almost 2 meters tall. He's a pretty big guy, and that rock is almost 3 times his size. So you can see it's a pretty massive disturbance event caused by the earthquake. And here, just after the earthquake, when we were finally able to get in the subtidal zone, we quantified how much large brown algae was left. And so Waipapa Bay, which is that images we just showed you that were 6 meters of uplift, you see on the graph there, there's a too high for the degree of uplift. Waipapa Bay North or Waipapa Bay South, kind of 2 different sites because one of them had less algae than the other one, so then we can kind of say that's a bit of a different environment afterward. Compared to Okiwi Bay, which was about 500 meters south of Waipapa and had about 2.5 meters uplift, so that's call it a medium uplift. There was just about 20% of large brown algae after the earthquake. Compared to down in Kōrero Peninsula, which had less than a meter uplift, and we'll consider that low. And after the earthquake, there was probably about 50% algae, and that's probably pretty similar to what the subtidal environment has. In that part, so it's probably that would be considered a bit of a control, I guess, for this is what we can call it. The intertidal was hit pretty hard there, but because I was looking more at the subtidal environment, less than a meter uplift didn't cause that much of a disruption in the subtidal algal community there. When we look at these large brown algae adults, we know that they're fueled from these juveniles, which we were just talking about with Caitlin and Zoe. And so these juveniles are almost as important as the adults because you need them to turn into the adults. And then you need the adults to turn back into the juveniles. But to go from the adults to the juveniles, there's a bit of a bottleneck. And so that life history part here is really important to try and perceive because without that, we don't have the adults. That's quite simple population dynamics, I guess. And some of the main causes of this bottleneck is you think about these little tiny spores or these propagules or a lot of different things you can call them, but they have to be spread out by their adults, land in a really, really nice area on a reef that's solid and won't move around. And then once they land in that spot, they have to actually attach to the seafloor. And then after they attach to the seafloor, as you'll see a little later on, they might have to then form another form of reproduction to turn into another thing before they can actually even turn into a sporophyte. And this whole time, they can't be eaten, they can't be smothered, they can't be destroyed in any way, or else that's it. And so in some cases, there's 0.0001% survival of some species. And so we wonder, can we then take these, can we loosen this bottleneck by kind of enhancing more of these juvenile algae and then start to, tip the balance back towards our large adults, which then can then reseed naturally and help out the system. And so to do this, we have to consider some things I've talked about a little bit, but life cycles are important. So you see kelp here compared to fucoids. Fucoids, you can see that there's a little bit of a difference happening in their life cycles here. Fucoids have more of a direct kind of propagule release where they have an egg and a sperm that turns into a zygote, and then that can grow into an adult. Compared to the kelp, you have this alternate generation happening where you have your zoospores that come out of your adult, turn into a male and a female gametophyte, and those gametophytes have to attach, and then they have to find each other and then turn into sporophytes later on. And so you can see here where the kelp is a bit trickier. Usually the kelp have more fecund, so they can spread out more spores, so they can kind of beat this alternate generation. But when you have a situation like the Kōrero earthquake, where there's a lot of gravel moving around afterward, and it's a really dynamic environment, likely the kelp are going to have a really tough time trying to recover into these spaces. Then you have to worry about different reproductive periods. So you have here a couple of fucoids, Landsbergia, Marginariella, Duvilia, the big bull kelps, and Lawsonia, the kelp that I looked at, all reproduce in winter, early spring, compared to some of these other fucoids like carpophyllums, sargassums, and cystophoras, which reproduce in spring and summer. So here you have totally different reproductive times. So say when an earthquake happens in November, it's going to affect how things recover, because it potentially the whole last period of reproduction could have all been ruined because of that earthquake that happened in November. And it's also likely that these different reproductive periods, then the juveniles will respond differently to heat waves, sedimentation, light attenuation, because they're more resilient to different things depending on when they actually would be growing on the reef. And so one of the things I did in my PhD was look at this in a lab to kind of get more of an idea, more of a scientific basis to be able to talk about these different stressors. And so I set up this lab experiment by taking a bunch of these, 2 fucoids and a kelp from the Kōrero coast. Kind of what Zoe was talking about is you can grab these reproductive bits of these algae. I did mostly intertidal stuff, or I got them as beach casts, so I didn't have to actually go and like remove them from the already degraded environment. And you can capture them when they get pushed up on the beach, bring them in, and then you can make this kind of slurry, and then you can dump it on these plates, and you hope for some good stuff to come through, some growth to happen. And then you set up this really incredible experiment, and you're like, oh, I'm going to figure everything out here. And you can see I have 3 different temperatures, 6 different treatments of light and temperature. And it was it was quite an endeavor. And looking back on it, I wasn't quite sure what I was getting myself into, but it was under 100 days that I watched these things grow and die, and every day I had to add sediment, and it was quite enlightening and pretty informative, I think, because we found pretty species-specific things that happened here. And this recently just got accepted, or recently got published in Marine Ecology Progress Series. So if anybody's interested in reading it, it was pretty good. But with Durvillia, we had pretty good growth at the control, so 11 degrees. Same thing with Lawsonia. But then when you added sediment, there was a little less growth. But then at 17 degrees for both of these species, there was pretty low growth and pretty low survival, no matter if it was a control or a high sediment. But the high sediment always had the worst effect on these juvenile species. Compared to Landsbergia, which is an oak leaf type fucoid. If anybody's been diving out there, it's kind of a stringy type seaweed. It has these little beautiful oak leaves on it, and it's a gorgeous seaweed. But you can see here in the control for 11 to 17 degrees, it grew pretty well, and the 17 degrees actually had the best growth. And even in the high sediment, there wasn't a lot of growth, but it had the best survival. And interestingly enough, it also is the only species that really survived at Waipapa Bay. That was the place that had over 6 meters of uplift. And now almost, I guess it would be 7 years on from now, it's probably one of the only species that's actually really been able to persist throughout that whole time. So it's pretty interesting to see this happen in the lab experiment in 2019, and to see how things are going out there right now. It's there's a really nice correlation happening there. But then we're like, oh, well, essentially we were able to grow them in the lab, and we did some experiments on them, but can we actually transplant them back out into the reef environment? Because that's one of the things we're worried about here is, well, can we instigate the reproduction of some of these seaweeds that have been lost in these areas? And so is there going to be species-specific success in terms of outplanting? Does the age of the recruit, so if we keep them in the lab longer before we put them out, does that matter? Does the environment actually influence some of our success? And does grazing have an effect? So just some of the key questions that Caitlin and Zoe have talked about a little bit, but these are questions a lot of people are interested in because they're kind of these predominant stressors or just natural phenomena that happen. And so we're trying to get a better idea of what's actually going on, and to see if outplanting or growing might have a better effect for some species or the other. And so then I grew some more of these seaweeds in the lab, and then went out to Kōrero and put them on these frames. And we found that Landsbergia was still the best one. It was still the one that grew the best. But you can see here between A and B, after we outplanted them, this is within 4 days. And so an interesting thing that happened is that they bleached. So they went from being really brown to being almost white. And this is something I think is an important consideration for restoration, in terms of you're going from a really nice lab environment, and then you're putting them into
really hostile reef environment. And even here at the Kōrero coast, where there's pretty high sedimentation after the earthquake, that it was that much of a change in light, say at high noon, that it actually caused these pigments to bleach. So you think this is a stressor that would be good to mitigate in the future in terms of trying to help your algae survive. But we did get decent survival. We had it essentially, if you do the math, it was better than natural recruitment. But the problem is that on the Kōrero coast, we had such crazy movements of sediment that all of my frames ended up getting covered in sediment. There was one cellar that came in that it was over a meter of sand that moved in. And so it just covered these frames that were half a meter off the seafloor. And so it's just, it's a difficult place to work. And so then I moved to the Queen Charlotte's Sound to try and make things a little easier on myself. And then I put cages out and grew some more large brown algae. And here I did different ones because different ones grow here. I grew Macrocystis, Eclonia, and Cystophora, and essentially outplanted them along the different gradients I talked about earlier, entering mid and outer Queen Charlotte Sound. And found Cystophora to have the best growth, which is a fucoid compared to a kelp. And I also found that there was quite a bit of grazing action on my plates. And so it was quite informative to see that my, when I put the tiles in cages, they actually did have better growth compared to when there wasn't a cage and there was gastropods mainly that were able to get on there and graze them away. And so far, I guess just kind of moving into what we've done here at Kōrero and kind of some of the movement forward we have is we've had successful propagular sediment growth of 7 species, 4 of them fucoids and 3 of them kelp. And so we'll try and enhance that in the future, see if we can increase that sediment list and find some optimal laboratory practices, because a lot of these species, there isn't a lot of literature or much out there to say whether or not there's been success with some of these fucoids in particular. We're trying to nail down this early life stage here where we can really get things to settle and grow at the microscopic level and then start to take them into a more of a realistic restoration approach, I guess. And then we wonder, is there an optimal substrate? So I use mostly plates, these cement fiber plates, and I got pretty good growth on them. But you know, a lot of people use lines mostly for kelp. But we wonder, is the line even an option for fucoids? They have a different kind of settlement, hold fast structure. And so it might not be anything that's feasible. So we and Caitlin both talked about the green gravel approach. That's done pretty well with kelps. Would it work well for fucoids? Especially like, you know, putting things on gravel and dumping them on the Kōrero coast, I think would be a poor choice because I just saw frames that got covered in a meter of sand in a weekend. So likely gravel wouldn't do very well. But these are things you kind of learn about your site. And it's good site selection methodology. Then you also wonder, can you use things like, you know, oyster and green shell mussels as a settlement substrate? And then essentially you kind of give back because you're not actually putting anything that wasn't there before, like string, into the natural environment. And then you wonder, is that the size make a difference? You know, green gravel, you have this gravel that's really small and you can probably get a lot of different replicates and likely get a lot more growth on all of the gravel. But if you use big boulders, say like on the Kōrero coast, if you were to inoculate a giant boulder and just set it there, would you have a better chance for it to grow up to be an adult? And then once it's an adult, it has a better chance to then reproduce to give back to the natural environment. Different textures. So you think about sandstone compared to mudstone. These propagules have to settle, attach, like I said, and then once they attach, they have to not erode away. So some of these, you know, rocks are easier to erode or better, they have better kind of inner matrix for things to grow on. And then there's kind of the antithesis to what I was talking about with, say, green shell mussels. There's different fiberglasses that potentially you could use and grow on, and they might be better for aquaculture. So you can kind of start taking this into a different avenue to say, well, maybe we use fiberglass or we test it to see potentially that might be a better use for aquaculture compared to string in the future. And then algal density is a big influencer. So you have really high density, it might not actually be a good thing because all of a sudden then all of these little germlings,
competing, are trying to outcompete each other. And then when you put it out there onto the reef, all of a sudden one wave comes through and pulls them all off because all of their holdfasts are interlaced with each other. And then as Caitlin had this slide up earlier, she beat me to the punch, but I think this is a really important thing in terms of there's a lot of techniques that we can use towards active restoration. And I think it's important to start harnessing the ones that are more unique to your environment. And it's really, really important to then figure out which technique you're going to use, but then also realizing that there's probably a greater holistic change that needs to happen if you want to do this. And that was an important point that she had with her graph from Kelsey Miller that she made with her publication, is that, you know, you can put all this energy into things, but it's probably just going to you're going to have to keep fighting the same fights if you don't figure out what's going on behind the scenes and actually try and restore the, you know, mountains to sea type of idea what Zoe is talking about, and figure that out before we just start throwing buckets of money and resource away and then going back to the office and banging our head against the wall. And I think that's a thing that everyone's starting to figure out, and we're trying our best to realize how we can reduce some of these more chronic stressors. And then just kind of a quick shot that, you know, we likely to use some of these techniques to enhance aquaculture as well. My main objective is restoration, but, you know, you can use some of these techniques as an aquaculture, which then provides all kind of potentially persistent benefits like carbon sequestration, like Caitlin was talking about, but also you can provide resource to human populations that are on coastlines, likely in a better way than some land-based agricultures. So that's me for now, and just wanted to say thanks to a bunch of different people and organizations and everyone who's been involved. And yeah, thank you to Caitlin and Zoe and Allison and Sarah behind the scenes.
Kia ora, thanks, Dan. That was terrific. I just have to say there is so much to think about. So much to think about in a really good way, but it boggles my mind, the complexity of everything. Just for a start, the complexity of being a seaweed, trying to establish and grow up and then make future generations of seaweeds. Then we've got the complexity of all the things that we are doing in the world that are impacting our seaweeds. And then the complexity of going, okay, let's restore seaweeds. And you realize that's not straightforward either. But I'm energized by this rather than overwhelmed by it. So thank you, everyone. We have some great questions. So let's get to those. A point, by the way, because we started late, we will run a little past 8:30. So just be aware of that. So Candice asks, what might the various reasons be for a reduction in kelp forests? And is it a sort of a multi-situation sort of thing? Who would like to answer that?
I can sort of tackle that one. And I think Dan sort of already touched on what the main stressors for kelp are. And again, it will vary regionally. Kelp are a cool water species. So the increases that we're seeing in temperature as well as the increase in heat waves event are fairly detrimental to certain species of kelp in certain regions. Sedimentation and that attenuation of light are a really big one for New Zealand and other sort of developed coastlines. Overfishing and putting that ecosystem out of balance and having overgrazing by kinna is another big cause of decline. So there are many, and these things don't act individually, and they interact on different levels. And so it does make things more complicated, but not, like you said, that there's still definitely ways to tackle restoration and conservation and protection. And one of the big things is that if we focus on protecting the ecosystem as a whole, then that makes it more resilient to any of the further stressors that it's going to be exposed to. So such as climate change and all those things associated. So if we can focus on making a resilient ecosystem, then that's sort of, I think, the best approach rather than focusing on just kelp itself.
Yes, it does seem to me that climate change is a big added new stressor that really the seaweeds could do without.
Definitely.
Peter, from Peter, a question for Zoe. What would your takeaway lessons be for building interest and understanding in groups that are not directly connected with an environment that you're clearly passionate about? What works best to attract attention and build interest?
I have to say probably one of the most popular events was when we actually invited people to come and have seaweed cocktails. It really seemed to get people out, like thinking about seaweed in another way.
Tell us a little more about seaweed cocktails, just as a by the by.
No, we had a when we received some of the funding for the Lovely Murimu Regeneration Project, we had a cocktail like fundraiser and launch evening, and a local seaweed farm donated some seaweed, and we challenged local cocktail bars to make signature seaweed cocktails, and they were delicious but quite strong, so that was slightly problematic. But I think there's a lot of really creative ways that you can engage, like actually art and creativity, food, drink, conversation. We talk a bit about doing the seaweed dance at Lovely Murimu. So kind of like trying to like woo people in so they don't really realize what they're getting involved with until it's too late. So I think there are lots of really fun ways that people can engage that don't mean they necessarily have to get into an environment that might feel they might feel a bit unsure about. But usually if we've had a cocktail with them, then we can convince them that the next thing they should do when they've sobered up is to come and have a snorkel with us as well. So I'm always trying to get people into the ocean at the end of the conversation.
Ice cream would work well as a non-alcoholic version, because ice cream, of course, I don't know if it does these days, but famously used to have seaweed in it.
Oh, and it still does. Lots of different food products use seaweed in them.
Yeah.
But I've always thought of like a salted seaweed chocolate would be really great, like a salted caramel.
Yeah.
I'm going to patent that though.
Well, this kind of answers Ella's question, because she had a question, which is, what do school students do with the seaweed they investigate? Do they grow it at their school? Do they eat it? So clearly they do.
A bit of both. We do some gathering and use those for kai sessions where they're cooked and used in all sorts of different ways. So one of our terms is just about innovative uses for seaweed, and also looking at traditional uses, right? It was a really important food source as well. But yeah, the second part of that question was around the students.
Do they grow it at their school?
We've tried a few times. Dan will know because he's obviously growing seaweed. It's very difficult to maintain a really like top-notch aquaculture system to get your seaweed growing. You know, diatoms, if you've got a little herbivore in there, that's the end of it all. It's quite hard to keep the water clean. So we tried a few times with the students, but they weren't as sort of detailed about caring for the tanks as they needed to be. So we had a few dead seaweed. So they help collect the spores, they help do that bit of the work, and then we take them back to the lab at NIWA and we get the aquaculturists there to take care of them and do little diary logs back to the school about how their seaweeds are doing.
That's a nice way of solving that problem.
Yeah.
So here's a question for Dan, which is kind of a bit, are the seaweed spores, the reproductive cells, released at a particular time? Is that related to the tide and moon? So you guys have said that things often happen in winter, but are there more particular timings involved with that?
I think there is some evidence that there is particular timings. I know with bull kelp, the idea is, because you think about, there has to be some way where they release at a particular time when it's calm, and then they can fall straight to the seafloor. But in general, I think there's kind of a continuous release that happens over the reproductive season. So that's one of the things that people have looked at, is if you take a particular plant and follow it through a season, will its spores or its propagules increase or decrease through time, because you're constantly producing them and then using them. And it's kind of like this I guess conveyor belt. But you also wonder then which is the best part of the season to actually collect them. So that's one thing people are trying to learn as well, is if you collect them early in the season compared to end of the season, does it actually influence your success of stock as a seaweed later on. I think that there is some evidence to show that there is. Usually if it's a settled period, but a lot of these, especially the kelp, still, I mean, they reproduce like a macrocystis. You can collect it in the winter all the way up until late spring, so they're definitely not releasing at only one time, because there's this continual thing happening that as they release, they'll get more and they'll get more and release.
It strikes me that there's quite big differences between the kelps that you've been talking about, Caitlin up in the Hauraki Gulf, Zoe in Wellington, Dan down on the Kaikoura coast, that those different species and those different ecosystems all have their own individual challenges. If you are thinking about how can we maximize the health of our kelp forests and maybe help restore them if they need restoring, do you think there's a one-size-fits-all package?
I don't think so. I mean, you just look at the different beech trees, if you think about a forest where there's shrubs, there's big trees, there's a whole canopy, understory, everything happening, and they all have to work as one unit. And in each one of these ecosystems, ecotones you talk about on a coastal environment or in a forest, all have their different unique things they do to then take advantage of that situation. And things change, and it's kind of the study, the whole, what ecology is. Things do change, and you have pioneer species that turn into kind of the next step of succession, next step, and then eventually you get to, an old growth forest. But things do change, and we're not adverse to that. We just, when things change really fast, and the organisms that are there can't actually catch up, that's usually where you get into these, tipping point scenarios, likely that, when Kina barrens happen, and then all of a sudden you can't get back to any other, I guess, healthy environment, because you kind of get in this nasty stasis of degradation.
Your mention of canopy-forming reminds me, there was a comment in the chat from Ian, basically paying tribute to Roger Grace, who did a lot of that early work in the Hauraki Gulf, teasing out what was causing the canopy-forming. A question for Zoe in particular from Mero: how can one get involved with the kelpers?
Yeah, well, we're just doing our pilots now, and when our plant outs go out, that will be when our kelpers, we're doing a bit of baseline survey around some of that work, but we plan to run another training program probably in September now, when our freedive instructor is back down again. But people can go on through to the website and follow up and get catch ups, and that's where we kind of communicate with people about when those opportunities arise. But yeah, we've just trained 8 community members and 8 of our older students, but our hope is that actually we build a really good community through that, and we're building a training course that sits alongside that so that they can feel really assured they've got the skills they need to go out and do it. So if they can, get in touch.
Terrific. I've got a question from Marina, who says, what do you think of restorative mariculture? I didn't know that as a word, mariculture, that increases ecosystem protection by also providing economical growth. Thoughts from any of you?
Well, I think there's talk about that. I think some of Caitlin's work is looking into that as well, to see what kind of services a kelp can provide, or a seaweed. And the idea is that, if you're getting economic value out of these organisms by growing them, you can also potentially create a service such as carbon sequestration. And there's also the idea we potentially, take out nutrients in a eutrophic environment. So if you have them next to a river outfall that's coming out of a city center or something, you might be able to decrease some of the eutrophication that's happening in a coastal environment, and potentially help out that way, create better water clarity. I think there's still some evidence needs to be shown for this to be really productive. The carbon sequestration part of it, I mean, Caitlin probably has more of an idea with this, but the problem is that once they're grown into the seaweed, if they're then taken out and then used for things that aren't, say, put into a home or dumped into the ocean, then you don't actually remove the carbon, and that can cause a bit of a a dilemma in terms of your carbon sequestration activities. But I think there is interest in this, and people are looking into this quite intensively.
Apai, well, I think to close off tonight, I've got a question. We'll go around the group in the order in which you spoke. And it's, what does a healthy reef look like in your area? And what are the first steps, the most important steps you recommend we take to look after these rocky reef ecosystems? What do you reckon, Caitlin, up in the Hauraki Gulf?
I think the picture of health of an ecosystem varies, but when I jump into the water and go through healthy kelp forests, I want to see the biodiversity. I want to see the connections between the organisms. I don't want to jump in and see something that looks barren. I want to see complexity. So that's, I think, to me, what it might be different species depending on, which reef you're on, which region you're in. But if there's complexity, if there's biodiversity, if there's, functioning connectivity in that system, then that's a pretty happy system to me.
Sounds great. What about you, Zoe?
Well, we're really lucky, right? Because in Wellington, you can step off into Tapaturanga Marine Reserve and you're in the most incredible seaweed forest there. So Wellington is really lucky. Our south coast actually has hundreds of different species of seaweeds, and it's abundant and diverse and beautiful. It's like flying. But that's contrasted, of course, by when you go into the harbour and you go, well, this is where all these problems are really concentrated. So in Wellington, we get to see that when we go snorkeling inside the marine reserve. So yeah, if I need to go see a healthy reef and get inspired for, the harbour will look quite different. It's a different environment, so we'll have different species that are abundant in those places. But just as Caitlin said, diversity, abundance, and I think you also you feel it in yourself when you're in those environments and you're snorkeling through. There's another kind of experience that's had.
Yeah, the Tapaturanga Marine Reserve is a wonderful resource. Dan, What about a healthy reef for you?
Oh, kia ora, Alison. This is Zoe. I remember when I was at Busel and I could go right off of the south coast on a nice day. It was so beautiful. You just essentially walk right into it, and it's amazing. I know that just doing a lot of that Kōrero earthquake subtidal work, where when you're down in the peninsula and you're swimming around and you have a decent water clarity day, and you'd be underwater doing some of these surveys for almost 2 hours because you had to look underneath everything. You had to actually swim up, look underneath Eclonia, look underneath Lawsonia. When you're underneath that, you'd see this whole suite of red foliose algae. Then there would be understory brown algae that would be growing there. And then on the bottom layer, you'd have encrustine and turfine, and you'd have this whole 4 different layers of seaweed that are all creating this underwater forest, which I guess is the whole idea of what this webinar is all about. And you talk that kind of thing when you're there, and you turn around, you have blue cod and blue moki and red moki, and sometimes you get a butterfish to swim by you, and then all of a sudden you look on the other side and you see a kina crawl by, and then you have an octopus that swims out. And it's kind of the idea of biodiversity that Caitlin's talking about, but it's the same thing when you're in a forest and you walk through and you have these different layers of things that are happening. And without that, you don't create that rich, diverse community. And to me, that's what I would like to see when I'm underwater.
Apai, there's nothing quite like swimming or snorkeling in a kelp forest. And it really is, as Zoe said, it's like flying. You're flying through the canopy of the forest. Anyway, a huge thank you to tonight's speakers. You were all wonderful and very inspiring. To you, our audience out there, thanks for being here with us and for asking such great questions. I'm very sorry if we ran out of time before we got to your questions, and I'm also sorry that we had some technical difficulties at the beginning and started late. Apologies about that. Kia ora. Many thanks to Sarah, working away behind the scenes to make all this happen. If you can just take a minute before you run off, please fill out our survey, which we've just posted in the chat. We'll also send you a follow-up email, which will have this link, because we really like hearing what you thought about the webinar so we can make the next ones even better. Don't forget, if you missed our previous webinars, they are all online. You can find them on the SeaWeek website via the news tab, or you can also look for SeaWeek's YouTube channel or Facebook page. They're there under the videos section. And stay tuned for news of future webinars. A karakia to close before we go. Ona hia, ona hia, ona hia ki te uru tapunui, kia wātia, kia mama, te nākau, te tinana. Te wairoa, te ara takata, koiara e rongo, whakairia aki ke ronga, kia tina, tina, huie, taikie. Remember, ko o te moana, ko te moana, ko o, I am the sea and the sea is me. Thanks for your company, everyone. Ata marie, good night. Stay safe.